In an age when federal money for scientific research is dwindling, the Salk Institute for Biological Studies recently struck gold with a $42 million grant. Already, the Helmsley Center for Genomic Medicine at Salk is getting promising results for a new cancer drug, which really isn't so new at all. Reuben Shaw, Associate Professor at Salk, joins me to talk about the research. Professor Shaw, you made a link, a discovery nearly a decade ago between lung cancer and a common diabetes drug known as fenformin. What was the link?
So the link was in trying to understand what this lung cancer gene was doing, we discovered that the major function it has in the cell normally is to actually regulate cellular metabolism. And it acts as a kind of fuel gauge to tell the cell how much energy it has. In tumors that actually acquire alterations in this gene, then they lack the ability to know how much energy they have. Tumors normally have plenty of energy and plenty of blood supply, they're fine, but it suggested a potential therapeutic Achilles heel, if you will, for tumors that have this particular genetic alteration. So then searching for what types of things might lower the metabolism of cells, it turns out that they're not cancer drugs, existing cancer drugs, but actually the world of diabetes research. So the most commonly used diabetes drug is a drug called metformin. So we started exploring metformin and analogs of metformin that are more potent to see whether or not they might have the ability to selectively kill off the tumor cells that lacked this gene. And indeed, that was the basis for this study.
Okay, so you and a team of other scientists did this study, and we have some graphics of the results. Can you talk to me about those?
Yes, so on the left-hand side, what we see, we treated a series of genetically engineered models of lung cancer that carry alterations in different cancer genes. And on the left-hand side, we see tumors treated with placebo as compared to, on the right-hand side, lung tumors with this particular genetic alteration treated with fenformin. And what we observed was a shrinking and a killing off of the tumor cells that lack this particular gene when you treat with fenformin. For lung cancers that have alterations in other genes, the fenformin was not effective.
I want to talk about that. So, I mean, as we all saw, the results are pretty dramatic. What emerges from this is what you call specialized treatment. Tell me about that.
Yeah. Personalized. This ties in with the idea of personalized medicine, which is becoming more and more common in cancer therapies today. And the idea is that you treat different patients based on the genetic alterations in their tumors. So the subtype of lung cancer then gets treated based on which particular genes are altered. This has been done in other cancer types for about the past 10 years. So in breast cancer, you may have heard of something, people say, oh, my tumor, I heard from the doctor, is ER positive. ER is just a particular gene. If your tumor has that gene in high amounts of it, it means that you can use a therapy called tamoxifen to actually treat those breast cancers. If your breast cancer is ER negative and doesn't have it, well, then tamoxifen won't do anything for it.
And how far away is the treatment? What stage in the process are you?
Great question. In this particular case, because metformin is the most widely used diabetes therapeutic, and this more potent version of it, fenformin, has FDA approval and was used for diabetes for years, it can quickly be transitioned into the clinic. And so me and my team are already working with clinicians on the East Coast and in Montreal, actually, to translate these into phase 1 trials to see what doses might be effective and in combination with what other existing standard of care therapies.
And what might be helpful in your further research is a $42 million grant that Salk recently received, I believe the largest in the Institute's history.
That's correct.
What's it going to be used for? What's the money going to be used for?
So this money is going to be used to fuel research into what we call genomic medicine, which is really this underlying idea that all different diseases, not just cancer, but cancer, diabetes, Alzheimer's, have a genetic component, and that only in the past couple of years has the technology to sequence all of the genes in a diseased tissue as compared to a normal tissue, or in a patient that has the disease compared to a normal patient, been possible to gain the full genetic sequence and then tailor therapeutics based on that. So we believe, just as this this example with using a diabetes drug for cancer illustrates that there are far more connections and basic processes that go wrong in these diseases, which includes changes in metabolism, changes in inflammation, and this grant will be used to create genomic sequencing facilities and to create imaging facilities and other core infrastructure, as well as to actually help pay for these advanced experiments with teams with the goal to expressly translate this into the clinic.
Reuben Shaw, thank you so much for coming in today.
Thank you very much, Mita.